20.2.1 Structure of contractile proteins. Each actin, thin filament, is made of two F filamentous actins, helically wound to each other. Each F actin is a polymer of monomeric G globular actins. Two filaments of another protein, tropomyosin, also run close to the F actin throughout its length. A complex protein, troponin, is distributed at regular intervals on the tropomyosin. In the resting state, a subunit of troponin marks the active binding site for myosin on the actin filament. Figure 20.3a. Each myosin thick filament is also a polymerized protein. Many monomeric proteins called meromyosins, figure 20.3b, constitute one thick filament. Each meromyosin has two important parts, a globular head with a short arm and a tail, the former being called the heavy meromyosin HMM and the latter the light meromyosin LMM. The HMM component, that is the head and the short arm, projects outward at regular distance and angle from each other from the surface of the polymerized meromyosin filament and is known as cross arm. The globular head is an active ATPase enzyme and has binding site for ATP and active site for actin. 20.2.2 Mechanism of muscle contraction. Mechanism of muscle contraction is best explained by the sliding filament theory, which states that contraction of a muscle fiber takes place by the sliding of the thin filament over the thick filaments. Muscle contraction is initiated by a signal sent by the central nervous system (CNS) via a motor neuron. A motor neuron, along with the muscle fibers, connected to its constituent, a motor unit. The junction between a motor neuron and the sarcolemma of the muscle fiber is called the neuromuscular junction or motor end plane. A neural signal reaching this junction releases a neurotransmitter, acetylcholine, which generates an action potential in the sarcolemma. This spread through the muscle fiber and causes the release of calcium ion into the sarcoplasm. Increase in calcium ion level lead to the binding of calcium with a subunit of troponin on actin filament and thereby remove the masking of active sites for myosin. Utilizing the energy from ATP, hydrolyzes the myosin head now binds to the exposed active site on the actin to form a cross bridge. Figure 20.4. This pulls the attached actin filament towards the center of A band. The Z line attached to these actin are also pulled inward, thereby causing a shortening of the sarcomere, that is, contraction. It is clear from the above steps that during shortening of the muscle, that is, contraction, the I bands get reduced whereas the A bands retain the length. Figure 20.5. The myosin releases the ADP and P goes back to its relaxed state. A new ATP binds and the cross bridge is broken. Figure 20.4. The ATP is again hydrolyzed by the myosin head and the cycle of cross bridge formation and breakage is repeated, causing further sliding. The process continues till the calcium ions are pumped back to the sarcoplasmic cisternae, resulting in the masking of actin filament. This causes the return of Z lines back to their original position, that is, relaxation. The reaction time of the fibers can vary in different muscles. Repeated activation of the muscles can lead to the accumulation of lactic acid due to anaerobic breakdown of glycogen in them, causing fatigue. Muscle contain a red-colored oxygen-storing pigment called myoglobin. Myoglobin content is high in some of the muscles, which gives a reddish appearance. Such muscles are called the red fibers. These muscles also contain plenty of mitochondria, which can utilize the large amount of oxygen stored in them for ATP production. These muscles, therefore, can also be called aerobic muscles. On the other hand, some of the muscles possesses very less quantity of myoglobin and therefore appear pale or whitish. These are the white fibers. Number of mitochondria are also few in them, but the amount of sarcoplasmic reticulum is high. They depend on anaerobic process for energy.